In 2018, millions of patients around the world were unknowingly taking a blood pressure medication that contained a probable human carcinogen. Not at some catastrophic level, but at a chronic, insidious level that had been building undetected for years. The molecule was NDMA, the drug was valsartan. And the reason nobody caught it earlier is the same reason your program might be at risk right now. The standard test one designed to find it. Hello, I'm Raghuvar Singh Parmar, the Director Scientific Affairs at ResolveMass Laboratories, and we work with pharma and biotech teams every day to identify nitrosamine impurities before regulators or patients find them. Today, here our nitrosamine testing series, we are going to start at the beginning what nitrosamines are, where they come from, and why they matter, and what a compliance-ready testing program actually looks like. Let me set the scene. In 2018, a routine inspection at a Chinese API manufacturer uncovered something that sent regulatory agencies into crisis mode. NDMA, a probable carcinogen, was present in valsartan at levels orders of magnitudes above the acceptable intake. The root cause was a seemingly routine solvent change, one that introduced a nitrosamine formation pathway nobody had modeled for. Valsartan was followed by losartan, irbesartan, then ranitidine and metformin. What looked like an isolated event became a systematic industry blind spot. And the uncomfortable truth is that every one of these recalls happened after patients had already been exposed. What exactly is a nitrosamine? So what are we actually dealing with? Nitrosamines are N-nitroso compounds formed when a secondary amine reacts with a nitrosetting agent under the right conditions of pH, temperature, and concentration. The IARC classifies many of them as probable or possible human carcinogens, and the mechanistic toxicology backs that up. They are DNA alkylating agents, meaning they can directly damage genetic material. The four names you'll hear most often in pharma are NDMA, NDEA, NMBA, and a newer category called NDSRIs, nitrosamine drug substance related impurities, which are unique to each API and represents the frontier of the current regulatory challenge. These are not interchangeable. Each has its own acceptable intake limit, its own analytical challenge, and its own formation story. Let's talk about how nitrosamines end up in the medicines, because the question that keeps CMC up at night, where are they actually coming from? The short answer is more places than most teams initially suspect. API synthesis is the most studied route. If your process uses secondary or tertiary amines and anything that can donate a nitrite or nitrous acid species, you have a potential formation event. But excipients matter too. Magnesium stearate, microcrystalline cellulose, povidone, common tablet excipients carry measurable nitrite levels. Nitrocellulose packaging coatings can introduce nitrosetting agents directly into contact with your product. Even processed water, if not carefully controlled, contributes. And then there is degradation. NDMA in ranitidine wasn't manufactured in it. It formed spontaneously in the product over time. Each of these pathways requires a different risk assessment, and a different analytical approach. 3 reasons this is not a simple checkbox exercise. Why does pharma care so much? Because the consequences stake. First, there is the patient safety dimension. NDMA is a group 2A probable human carcinogen, and chronic daily exposure, even at microgram level, can meaningfully elevate lifetime cancer risk in a population that is already taking the drug for the chronic conditions. Second, there is regulatory exposure. The FDA's 2020 guidance and the EMA's Article 31 referral procedure created legally enforceable expectations. Risk assessments are not optional for products found to exceed acceptable intakes. Batch testing before release is required. Third, and this is the one that gets executive attention, is supply chain disruption. A single out of specification nitrosamine result can trigger a market withdrawal, a product recall, and a CMC amendment that takes months to resolve. The valsartan, ranitidine, and metformin cases delivered all 3 consequences simultaneously. Which products are most at risk? Not every drug product carries equal risk, but no product class is fully exempt. That's the critical nuance. The highest risk category is the synthetic small molecule drugs containing secondary or tertiary amine groups in their structure. ARBs, ACE inhibitors, metformin, antihistamines, antidepressants. These molecules have the chemical architecture that can react with trace nitrite to form NDSRIs. Oral solid dosage products are statistically the most affected because the contact between the API and excipients over a shelf life of 2 to 3 years creates both the opportunity and the duration for formation. Complex formulation with reactive excipients sits in the middle tier. Biologics and sterile products are generally lower risk but not zero. Processed water quality and packaging interactions still matter. The pyramid isn't about exemption, it's about where to focus first. Real products, real consequences. The cases look different on the surface. Valsartan was a manufacturing process issue. Ranitidines NDMA was inherent to the molecule itself. It formed from degradation under normal storage conditions. Metformin's NMBA source is still debated, but the pattern underneath is identical. Detection came years, sometimes a decade after patients have already been exposed. At ResolveMass, we have supported sponsors through investigations that started with a single unexplained peak in the chromatogram and ended in a packaging changes, excipient substitutions, and refill submissions. The teams that built nitrosamine thinking into their development programs early found lower levels, had more options, and spent far less time in crisis management. The ones who waited for a recall notice had almost no good options left. 5-step nitrosamine compliance workflow. So let's talk about nitrosamine compliance workflow containing specific 5 steps. What does a compliant nitrosamine program actually look like? The FDA's September 2020 guidance and EMA's parallel requirements lay out a clear sequence. And requirements, it starts with the risk assessment, not testing. You map every potential formation pathway, synthesis, excipients, packaging, degradation, processing aids. You rank them by likelihood and consequences. Only then do you design the analytical methods, because the right method for an NDMA risk from synthesis is not the same method you'd use for an NDRI risk from API degradation. Confirmatory testing validates the method and establishes real-world levels. Limit evaluation compares what you found against the ICH M7 derived acceptable index. And finally, the regulatory story captures all of this in a format that survives FDA reviewers' first question. This is a life cycle, not a one-time event, and the agencies expect you to treat it that way. So what should you walk away with this discussion? Nitrosamines form from chemistry, not negligence. Any synthetic API program can harbor a formation risk. Standard HPLC will not find them. Sensitive LC-MS/MS or GC-MS/MS method is required. Regulators globally now require documented risk assessments. This is not an optional, and it applies to your drug product regardless of therapeutic area. And early proactive testing costs a fraction of what a post-recall response costs, measured in time, money, and patient trust. If you are working on an IND, an ANDA, or post-approval product, and you need a CRO partner who treats nitrosamine compliance as more than a tick box exercise, ResolveMass Laboratories supports small and mid-sized pharma teams from the first risk assessment all the way through confirmatory testing and regulatory submission. Thank you so much for watching this session until now, and see you in the next one. Goodbye for now.